Thanks a lot for this invitation to give a talk about the science that we are doing here in Tampere University in Finland. We are the happiest country in the world 5 times in a row. So I'm very happy with this fact and very happy to give a talk about my research. I have changed a bit the title, so don't worry. It's about the same thing, but it's just a bit different. So I will be talking about light-induced surfactant cleavage via by using far-red light via triplet sensitization. So let's move on. So at the moment, for photocleavage and thus, for example, photopolymerization or for cargo release, such as drug delivery systems, people have used a lot of photocleavable or photoprotecting groups. Initially, they have used ruthenium complexes, then ortho nitrobenzeols and coumarines are among the, I think, most famous ones. And then in time, people started to make new photocleavable moieties to expand the excitation wavelengths toward red by using, for example, porphyrinoids, phthalocyanines, and most recently by using body pie derivatives. The reason behind it is that body pie cages or body pie photocleavable moieties have high mole extinction coefficient, so they absorb light efficiently, though usually in green. But there are conjugates that may absorb light in red or near infrared. They are very resistant to photo bleaching, so they are very, very stable. And so far, the photolysis or photocleavage yield was rather modest, but still enough to do a lot of things. What they have noticed recently, I mean, Arthur Winter from Ohio State University and Peter Klan, that actually halogenation, adding bromine or iodine or chlorine groups here to the body pie moiety increases actually the photolysis quantum yield, so the photocleavage becomes more and more efficient. And here I show you the photo. It's a Jablonski diagram of this process. So they claim that it happens via both singlet state cation and triplet state cation. So we here in Tampere were questioned whether it's possible to use then triplet sensitization or triplet energy transfer. Thanks for nice talks about triplet chemistry from previous speakers. So what if we use triplet sensitization by using another molecule for photocleavage? Will it work, first of all? Unfortunately for us, this was published recently by a group of Professor Wei Ping Wang from Hong Kong University, where they introduced this approach. In their pioneering paper, they have used this platinum tetraphenyl tetrabenzaporphyrin and body pie conjugate of chlorambucil, anti-cancer drug. And what they have shown that by exciting this porphyrin with help of red 625 nanometers light, they were able to cleave this body pie conjugate without direct excitation of body pie core with green, which is usually happening with green. Then they moved on and they created this surfactant. Why? Because it's very tedious process to conjugate a body pie with every anti-cancer drug, and 90% of anti-cancer drugs are hydrophobic. Does it mean that you have to do the same routine every time to make a drug delivery system out of it? Of course not. So what they ended up with is that they introduced this surfactant molecule that can form micelle-like nanoparticles that can incorporate hydrophobic cargo molecules inside. And particularly, they can also upload, for example, sensitizer, similar to this platinum tetraphenyl tetrabenzaporphyrin. And upon either red or green light, they have noticed that there is a cleavage, so there is a disassembly of nanoparticles. So what has happened that step by step they cleaved 1, 2, and 3 body parts, and then cargo was released. And they did it also in vivo by using rodents. So they have used paclitaxel anti-cancer drug, and they have proven that triggering light triggering worked, and there was a shrinkage in tumor site, and etc. So this approach worked, but our idea was then to expand actually the excitation wavelengths towards far-red near-infrared light, because of the fact that it's known that the near-infrared light has highest possible tissue penetration, as you can see from this image. So we decided that would it be possible to use 750, 730 nanometers light instead of 650, 625 that previously was used. But there is an obstacle here, that if you want to shift the excitation light into far red, it will make energy transfer, which was the initial step of the photocleavage, endothermic. So it's not enthalpy driven, so it's not very efficient, should not be very efficient, and should be very, very slow. So there is a logical question whether it will work, I mean, for photocleavage, because enthalpy will not work for us. Luckily, we have entropy also, and it can contribute to overall free energy. So you can hear it from this equation. So entropy is here. And from previous works, we know that if you can do something with the triplet state of second molecule, like body pi in this case, that will accept triplet energy from our donor, for example, porphyrin, it should actually work despite of the fact that we are lacking enthalpy in this case. We did it for several other works about photon up conversion and switching. So we have had some preliminary data or ideas that it should actually work. And for that, we have used this already published surfactant that we have actually taken from Professor Wei Ping Wan laboratory in Hong Kong University. And we have used this commercial platinum tetra naphtha porphyrin, which has strong absorption in far red and up to even near infrared here as well. What is good about these two molecules that you can selectively excite this porphyrin without exciting body pi at all. So there will be no direct excitation of body pi in this case. So everything which will happen upon excitation of this porphyrinoid happens because of its excitation. So there is no overlap. And we started triplet energy transfer by using phosphorescence. So this is a phosphorescence band of our porphyrinoid at 895. And then we started to monitor quenching of this phosphorescence in respect to addition of body pi molecule. This is this experiment we have performed in DMSO. So here we have no nanoparticles yet. This is just to confirm that there is an option, or there is a possibility to have triplet energy transfer from our sensitizer to body pi, and we have noticed a gradual quenching of phosphorescence in respect to addition of body pi. We have used it to plot so-called Sternfallmer plot, and from this we have figured out that the rate of the process is rather slow, so it's almost 2 orders of magnitude slower than it should be in DMSO, meaning that this process is very, very endothermic, so it lacks enthalpy. In other words, it means that we have confirmed that the energy of triplet state of this porphyrinoid is lower than triplet energy of our body pi cage. But again, there is a question: if we make nanoparticles out of these molecules, will photocleavage take place upon excitation of this porphyrin at 730 nanometers, similarly to what has been introduced before. To monitor photocleavage, we have used Nile red fluorescence release marker. So this is very hydrophobic molecule with very bright, very high quantum yield. So it's very bright when it's in a hydrophobic environment. So basically, upon making nanoparticles, we have incorporated Nile red inside them together with the porphyrin, together with our platinum tetra naphtha porphyrin. We have also created anoxic or hypoxic conditions by using sodium sulfate, so it depletes oxygen according to this reaction. So when we performed our experiments, there was no oxygen there almost, and upon excitation of our porphyrin, we have noticed gradual decrease in time of Nile red, because once we have disassembled our nanoparticles, a Nile red was released in a water environment, in buffer, and it started to precipitate because it's hydrophobic, it doesn't like water, and when it precipitates, it has no fluorescence. So in time, we have noticed this gradual release of Nile red. So in 25 minutes of low power light, 170 milliwatts per square centimeter, we have seen more than 50% Nile red release. Of course, if you use higher power densities, you may get faster release or slower release, and the percentage will be higher, but this is just to represent the fact that approach works, while actually at ambient conditions, this red curve, there was basically no release, maybe something like within 10%, but definitely lower than at anoxic or hypoxic conditions that we have made by using sodium sulfate. Then, of course, we were curious whether this will work in cells. So we performed just uptake studies by using confocal microscopy. Here on the left, we have nanoparticles only. We have imaged them by using 561 nanometer laser just to excite our Nile red molecule selectively and see its fluorescence and how it accumulates inside the cells. We have used also a laser tracker just to see the membrane of the cells for better imaging. And here on the right, you see how they, laser tracker to monitor, to see lysosomes, of course. And here you see overlap of nanoparticles emission and laser tracker. So from this, you can make a conclusion that nanoparticles were uptaken and they were colocalized inside lysosomes, because there is a clear overlap between a laser tracker emission and nanoparticles emission. And it happens roughly in 1 hour. So after 1 hour, we have seen almost complete uptake of nanoparticles. These images we have done after 2 hours of incubation, but it could have been imaged even after 1 hour. We have used PC3 cancer cells that are usually very hypoxic. Then here you see the imaging after a light activation of nanoparticles. So on the top, for comparison, we have images of the cells treated with non-activated nanoparticles without illumination. So again, there is a clear overlap of emission from nanoparticles and laser tracker. So they are in lysosomes. But after activation of nanoparticles, emission of Nile red decreased substantially. And there is a reason behind it, because once our nanoparticles were disassembled, Nile red was released in aqueous environment and has basically a weak emission, and this can be seen from these images. So that's how it works, luckily, or how it worked and works. It's just hot from the oven, actually, data that we have produced just 1 week ago. So I'm very happy to see these results coming from this drug delivery system. And basically, I would like to draw a conclusion from this experimental data that, in fact, you can use endothermic triplet energy transfer for photocleavage of body pi-based photocages or photocleavable photoprotecting groups. And we are doing actually it for the very first time. Because of the fact that we are using endothermic triplet energy transfer, it allows us to already engage far-red 730 nanometers light, which is 1 nanometers red shift with respect to what previously was published by our collaborators. So it can allow you to perform a light-triggered drug release deeper in tissue with high spatial and temporal resolution. And I would like to thank you for your attention. I'm eager to listen to your comments and your questions. And I would like to thank team of the project and organizers. First of all, I would like to thank my no longer PhD student, UC Socorti, who is now in University of Texas in Austin in US, our collaborators who have made molecules for us, who has done cell studies and imaging, University of Hong Kong people who have made a molecule for us. So as you can see, students are leaving and in good universities to pursue their careers, postdocs. And if organizers allow me, I would like just to advertise a job alert in my group. I will start my own small group from 1st of September in Tampere University, thanks to Finnish academy, yes, Suomen Academy funding. So we will be making and breaking nano materials with help of red light together with University of Helsinki people. So please apply. Thank you so much.